Good afternoon. Well, this is a report of the final report of a collaborative study, a collaborative project, and it's our aim to explain what we have done during all this time, and with the funding of the CIBERNET. So we have a duty, and it's our duty to explain our experiences and our results regarding this project. So the main findings are present here in the form of summary. We have done a lot of work, so it's very difficult to summarize in a few features of the work done during all this time. And for this reason, we have done this presentation in English, written in English, and the main publications are presented here. But what I think is that probably is better, because it's an internal report, to give all this talk in Spanish, if I have the authorization and the permission of the audience and the moderator, if you don't mind. If you agree, then I will start by explaining what is our aim. Is the cellular and molecular analysis of tau seeding progression in animal and cellular models. And this work has been carried out by myself, José Antonio del Río Fernández from the IBEC, and Jesús Ávila de Grado from the Centro de Biología Molecular Severo Ochoa. And the main objectives are these ones. So the first one was the use of microfluidic devices with primary cultures of neurons, astrocytes, and mixed cells to know what happened in vitro with the process of seeding and spreading of tau. The second one, the objective 2, is the role of microglial cells, not only microglial cells, but also astrocytes and other glial cells in addition to neurons. The objective 3 is to analyze, to assess the specific differences in seeding and spreading of tau pathology by using animal models following intercellular inoculation in the hippocampus and in other places of tau-enriched homogeneates from pure cases of Alzheimer's disease and other tauopathies. The objective 4 is to analyze in vitro the changes in the activity of neuronal networks between the interconnected areas subjected to tau seeding. And the fifth and last is to determine the effect of seeding and spreading after the combined inoculation of tau and other abnormal proteins. So we will start by talking about the first. Sorry. I cannot go to the second one, to the next. ¿Me puede pasar la siguiente diapositiva? Vale, muy bien. Bien, el objetivo 1, por tanto, es el estudio del tau seeding y spreading utilizando unos instrumentos o unas preparaciones que son los microfluidics cámaras o las cámaras microfluidos, y en el que una parte de la cámara se sitúan las células. ¿Perdón? Okay, so in this camera we use the cells, and in the other camera the axons from this first camera go through these small and thin channels to the other place. So the axons are located here and the neurons are located in the other place here. So when we inject, for example, tau, abnormal tau, phosphorylated tau, or even tau in this compartment, then tau can be projected or can be retrograded and transported to the first compartment. So what we can see here is a very simple device that permits the study of retrograde progression of transport of tau to from the distal part of neuron to the cell body. And here are many examples of these cameras, these devices, and here the observation, the Western blot observation that tau goes to one place to another place. So this is a very easy, a very simple device to study this point. The other one is a bit more complicated, in which we have used two different types of neurons. One of them is cells come from the entorhinal cortex, and the other one cells from the hippocampus. And what we are trying to do is to know what kind of interaction exists between this kind of two different populations located in different compartments, in different cameras. So in one place we put tau in the entorhinal cortex, and we try to know whether this tau is transported through the axons to the other compartment in which we have located the hippocampal cells. And then here in these small sketches and these small in these Western blots, we can see that at the very beginning, entorhinal cortex tau is located in the entorhinal cortex, but then following some days, at 7th day, we can see that from the tau is also located in this part. So the other is a bit difficult feature, because it tries to demonstrate the kind of synchronicity between the different populations of cells. And we can see here, and we will see this point in more detail later on, that synchronicity is lost once abnormal tau go from the entorhinal cortex to the hippocampus. So we will go into later on to discuss this in more detail. Well, we have produced this work, the microfluid laboratory platforms, just to explain what kind of devices are we using, not only to study this particular treatments tau with different populations, but also at the combination with neurons and other cell types. So this kind of work is in progress, and one of them this has been already published, but other papers are in preparation. The next one, the objective 2, is the role of microglial cells in the propagation of the disease. This has been mainly carried out by the group of Jesus Avila, and here is just a review of the role of microglia in Alzheimer's disease in the context of tau pathology. The other one is another review that we publish in Progress in Neurobiology, trying to summarize what is the role of macrophagic and microglial complexity after neuronal injury, including neuronal degeneration. So this is some pictures observed in this or presented in this kind of review. Also, we have also prepared this mini review regarding the propagation of tau via extracellular vesicles, and this has been useful because it is based on the basis of this mini review. We have produced some results regarding the uptake of tau by macroglial cells, and also in the other one, the internalization of monomeric tau from extracellular monomeric tau internalization by astrocytes. So here are many examples of one example of this kind of internalization, and here is the demonstration by the use of Western blots and double leveling immunohistochemistry and confocal microscopy of this kind of internalization. So this is an important point, because we are always talking about neurons and tau uptake, etc. But many other cell types are also involved in all these disorders. And astrocytes and oligodendroglial cells and microglia are important participants in this kind of alterations. Regarding this objective also, we have produced some secondary papers in some way. One of them is to look at the difference between human and murine tau at the N-terminal end. And this is an important point, because you will see later that it not only depends, the tau seeding and spreading, on the strains of inoculated tau, but also on the kind of host tau. So it's very important to understand what is the difference between murine tau, for example, and human tau on primate, tau in this context. And here we have seen that the N-terminal sequence of tau is very important. It differs from mice to humans. This tau terminal is also important because this is an N-binding protein site. So regarding the possibility of interaction with other proteins and with the capacity of aggregation of this tau, it is important to understand and to know that it's not the same tau, murine tau, when compared with human tau. And here are further examples of this kind of all the differences. And here, for example, we can demonstrate that EB and binding proteins facilitates tau secretion, whereas the EB1 depletion decreases tau secretion. So this is an important segment of the tau protein. We will go to, well, is in preparation, these important aspects of the difference between the different strains of tau and the differences of tau among different species, and the comparison of these different types of strains in tau, in human tau, regarding Alzheimer's disease, peak disease, corticobasal degeneration, and other tauopathies, in line with the studies that are being carried out by the group of Coedert. And also is interesting that the group of Jesus Avila demonstrated or has discovered a new and specific human tau isoform that lacks these exons, the 13, and after exon 12, it is translated into another peptide comprising the 18 residues. So this is a new form of tau. Other teams have to validate this kind of discovery. So although we are talking always about the 8 isoforms of tau in the brain, the human brain, so mainly in the human brain, there are also other isoforms of tau which are less expressed in human brain. The objective 3 is to use animals, to use mice for the tau seeding and spreading using different strains of different tauopathies. We have reported in previous years, in the previous meeting at CIBERNED, these three papers in which we demonstrated seeding and spreading of tau using cortex samples. Then the relevance of host tau, the primary, or the first result suggesting that host tau was important in this aspect. And then another one, which is the involvement of oligodendrocytes in tau seeding and spreading when inoculated in the corpus, when tau is inoculated in the corpus callosum. Well, we have followed this kind of experiments, and here we have studied the capacity of tau seeding by following the inoculation of argyrophilic grain disease tau. This is a 4R tauopathy. So remember that it's just 4R tau what we are injecting in the brain, and compare with part is in some way part of Alzheimer's disease. It's a kind of Alzheimer's disease at the first stages in which there is only neurofibril tangle formation, but not amyloid deposit. So there is a kind of discussion on whether this is a new disease or is the first stages or represents the first stages of tau of Alzheimer's disease. So this is important to note, however, that there is no amyloid in any case, and this is a 4R tauopathy, whereas the other one, the part, is a 3R 4R tauopathy. And here is just what happens in the brain with AGD or part, and there is difference. So there is neurofibril tangle formation, and here is mainly grains in addition to some pretangles. And this is 3R and 4R, and this is 3R and 4R. So these are the different strains when comparing 4R plus 3R tauopathy, as in Alzheimer's disease or in part. And here is the AG D, which is a 4R tauopathy. So what is interesting is that the pattern of seeding and spreading when we are injecting these homogeneous sarcosylin soluble fractions into the brain, in the hippocampus of these mice, is that the pattern is very similar in both cases. So there are neurons, there are grains in different locations, and also when we inject in the corpus callosum, there is an involvement of the corpus callosum ipsilateral and later in the contralateral corpus callosum in both cases. So here, in addition to threads and fibers of the corpus callosum, we can see many oligodendroglial cells. Whilst in the case of part, for example, is just a disease with neuronal implication or involvement, but not glial involvement. And here another important point is that although here in part the deposits are 4R and 3R, so what is expected following the inoculation of 4R, 3R tauopathy, here we can see also 4R but 3R tau. So something happens here that when we are injecting sarcosylin soluble fractions of different diseases, 4R and 3R, or 3R and 4R tau, then the result in wild-type mice is that the product, the new tau, is maybe tau 3R or tau 4R. Here is the distribution to show the localization and distribution through time following 3 months of survival or 7 months of survival. And here is the same. This is EGT, this is part, and you can see that the pattern is more or less similar. And through time, with time, the tau spreads to the contralateral side. So it's very clear that this is an active process. And another important point is that by using confocal microscopy, we can see that there is an involvement of neurons, oligodendrocytes, but never astrocytes in these cases. And another important point is that these deposits of tau, these abnormal deposits of tau, colocalize active P38. So this is a kinase which phosphorylates tau. So this is an active process. Another example is this one, familial globular myopathy, linked to MAP mutations, but also in sporadic cases. So this is a very important work because we have studied several cases of globular glial tauopathy, and we have inoculated also these homogenates through to the brain of wild-type mice. And here is a summary of the result of the alterations present in globular glial tauopathy. This is a 4R tauopathy, and with involvement of neurons and glial cells. And glial cells show the presence of very typical glial inclusions in astrocytes and oligodendrocytes. What we have studied is that the kind of alterations that exist in astrocytes and in oligodendrocytes by using immunohistochemistry and also transcriptomics and followed by validation. So here we have seen that there is an enrichment of protein alterations other than those expected for tau. So there are many other proteins which are phosphorylated in the white matter and in the cerebral cortex in GGT patients. So this is a kind of protein of protein interactome and phosphoproteome in the frontal cortex, indicating that phosphorylation of these abnormal proteins may have some impact in other molecules. So we will discuss this in more detail afterwards. Then the inclusion, the inoculation of this abnormal tau produces a similar pattern in this animal. So this is almost the same. So there is a localization in neurons, in glial cells, but only in neurons in this case. This is 4R tau and 3R tau. The involvement of the corpus callosum results in an early alteration of myelin. For example, this PLP1 is a protein marker of myelin, and this is an alteration. These are small globules of PLP1 abnormal protein located. And here is what happens when we inoculate in the caudate and putamen. So the results are almost the same. The observation by using confocal microscopies are almost the same. And again, we see an active process because there is an involvement of different kinases. And here is not just for an example of what happens in familial GGT, but also in sporadic GGT and GGT due to another mutation of tau gene. Here is another work has been submitted for publication to molecular neurodegeneration, in which we have used different tau models, so different murine tau models. So one of them is wild type. The other one is P301S, which express human 4R tau with the mutation P301S, which is pathogenic and produce frontotemporal degeneration in human. The other one is anomazygous transgenic mice, which is knockout for murine tau, but express human tau, mainly 3R, but also 4R tau protein. So it's different what this animal from the other types of murine adult murine models, because in adult mice, what predominates is 4R tau or is unique to 4R tau, whereas 3R tau is least frequent or almost not expressed. And the other one is a knockout mouse for human and murine proteins. Well, we have used this kind of models in order to demonstrate or to analyze what happened when we change the host tau. We have used the same inoculum for all the animals, so use the same amount and the same location of these sarcosylin soluble fractions. And here is what the different models. Here is the P310S tau. Tau is located in a widely in the brain, in neurons, in glial cells, in many different regions. There is accompanied by different forms of tau, including tau, truncated tau, and this is accompanied by the activation of different tau kinases, including P38 and JAK kinases, for example. And here is the comparison with the other model of expressing human tau, is this one. And this one, you can see here that the amount of tau is less prevalent, less important, and with much lower levels than the other one, which express the mutant tau. And in addition, the deposits are different. So, for example, here we can see that PHF1 is expressed, but with the characteristics which are completely different from those in this one. And the same happens with the other one, with MC1. The other markers, as for example, ET8, are negative. And what is interesting here is that 3R is much more abundant than 4R tau. And the other important point is that the knockout mouse is negative in the hippocampus, in the dendritic gyrus for any form of tau. And here is the Western blot demonstration of this difference among these different types of tau. Wild type, P301S, the knockout, and the H tau. So here are the main differences. And you can see here the difference between the presence of 3 tau and 4R tau. Well, when we are injecting wild type, you know what happened. So we have demonstrated before what happened with this kind of deposits in other models. Here is by using Alzheimer. And an important point is, for example, this picture in which 3R and 4R tau are indicated. So, for example, 3 tau is much more abundant than 4R tau. And here is another point important in which when we inoculate the tau in the ipsilateral dentate gyrus, in the ipsilateral hippocampus of the gyrus, we observe the uptake of abnormal tau, but also this uptake is observed in the contralateral dentate gyrus. That means that tau is transported through the commissural pathway, the dendritic commissural pathway, which is only present in rodents and not in humans. So this is a good, beautiful example of transport through the axons. So this is the other model, the P310S, and here is almost the same, but with much more abundant amount of tau in this kind of deposits. And here is what happens when we inject into the brain of knockout. When we inoculate into the brain of knockout, there is not tau. This is just a few macrophages just in the site of injection. And in contrast, there is a large amount of different species of tau in animals with expressing human 4R and 3R tau. And here is interesting because what we have seen is that 3R is present, but 4R tau predominates in these animals. The deposits are 4R, mainly 4R tau. When this animal, in normal conditions, what shows is an increase in the presence of 3R tau. Here is well, just to demonstrate that the kinases are colocalizing with the tau deposits in these animals. And this is to compare what happens, the difference between P301S and each tau, regarding the difference between the deposits in both types of animals. And here in this one, for example, we can see that the amount of tau is much higher in this mice expressing human large amount of tau than in those animals expressing lower amounts of mutated tau, mutant human tau. And this is also important because other kinases are also expressed in this animal. In addition to the previous one, the STEP kinases, the jump kinases on P58, and others are also involved. For example, GSK, AKT, PK. So here is an recruitment, an activation, many more kinases. And here is a summary of the difference between or among these different types of animals. And what is very clear is that the kind of deposits following exactly the same inoculum into the same region of the brain produce different types of deposits. These are the main important points. So, and the main new points, not only neurons are affected, but also oligodendrocytes. Spreading is time dependent. We already know that, and this is via neuronal connectivity and oligodendroglial contact. This is just an activation of local kinases, and there is also a difference between the kind of tau, the type of tau of the host tau. Also, it's very important that we are not producing only a change in the characteristics, in the post-translational characteristics of tau, but also in the translational characteristics of tau. So we are changing the expression of ratios of 3R versus 4R, or the contrary, when we inject some particular points of strains of host tau. This is a very interesting point because this is the first time that we can demonstrate that tau seeding can spreading produce changes, very important changes in the characteristics of tau, depending on the type of tau of the host. And when we consider that there is the possibility in the human brain that we have different strains of tau, not only regarding the diseases, but regarding the cell populations. So that means that perhaps not all neurons have the same levels of 3R, 4R, or many other species of tau. That could explain why some cells or cell populations are more vulnerable than others to tauopathies. And interestingly, in another study, which was not related to this one, the group of Jesus Avila demonstrated that following middle cerebral artery occlusion, permanent middle cerebral artery occlusion in rats and mice, then the oligodendroglial cells, which are located around the site of infarction, then show a shift in the expression of 4R versus 3R tau. So this is another model, completely different model, in which certain conditions, the splicing of tau is modified under pathological conditions. This is the objective 4, and this was to analyze the changes in the activity of neuronal networks between interconnected cells. So these have, well, the devices that I have shown at the beginning of the talk are these ones, but here what we have used is the study of movements of calcium. So to know the activity of these cultures of the cell types, and here is when this is a video, we could see the movement of this kind of a small white dots, and what happens when they are in normal conditions. So there is a kind of interactions between these cells and these ones, indicating that there is some kind of contact and interaction between these different populations, and it's feasible to study because of we have used in this experiment different softwares. Well, what happens when we put some tau in this kind of cultures? Here's internal cortex, the hippocampus, so at the beginning is nothing. Here we put pathologic tau, and then we have put we then carved out the calcium analysis, this one, and we can see here the different pulses, and we can see that following tau inoculation, all this system fails. So we have failed to in some way, because we have demonstrated that tau has the capacity of tau seeding, and the inoculation has the capacity of tau seeding and spreading in animals. Then when we use this kind of devices, what happens is that cells die. So we have not been able to study at this level the differences.
Isidro,
finally,
I'm sorry to interrupt. Yeah, you have to be finishing in a couple of minutes.
Yes,
please.
I will finish.
Thank you.
The other one is the objective 5, in which we have tried to study the combination of different molecules. On the one hand, including tau, and on the other hand, including alpha-synuclein, because we tried to study the comorbidities that are present in human pathology. So this is the previous work that was carried out using sonicated alpha-synuclein fibrils, and we can reproduce this kind of alterations following inoculation of these fibrils. So it's the brain of a mouse injected, and here you can see the fibrils in neurons. And here we have used another model by using virus expressed in different parts of alpha-synuclein. Here the system works perfectly when we are using alpha-synuclein segments of artificial alpha-synuclein, but we failed in the demonstration of that the homogenates of coming from multiple system atrophy and diffuse Lewy body disease or dementia with Lewy body cases failed in the appearance of this kind of thing. So we have not continued progress in this kind of combination because we failed in the demonstration of this kind of things. Well, and this has served to go to another protein, is the prion protein, and we have seen that the prion protein does not only interact with beta amyloid, but is also very useful in the regulation of tau, because PRP regulates tau, and also tau regulates the production of PRP protein. So there is another partner in all these kind of processes in which PRP plays a role in the capacity of seeding and and spreading of tau in different models. So this is the participation of different molecules in this kind of models. And I would like to finish here only to say that the COVID has produced some impairment and delay in the results, but we have, and just is 1 minute, to show that we have done other kind of studies in the meantime. So what we have demonstrated is that wet matter Alzheimer's disease produces alterations in the wet matter in oligodendroglial cells and astrocytes. And also a very important point is that the regulated protein phosphorylation is a determining condition in the continuum of brain aging and Alzheimer's disease. And in addition to, just to summarize, in addition to tau and alpha and beta amyloid, we see that at the very beginning and the first stages of the disease, not only the entorhinal cortex show abnormal protein phosphorylation or the regulation of protein phosphorylation, but also the frontal cortex. So this is very important, the regulation of proteins, including kinases. So the scenario, the basic scenario, is not only tau, the study of tau of beta amyloid, but also the many other proteins which affect the membranes, the cytoskeleton, and synapses, and many other pathways in the central nervous system. And it serves also to demonstrate that membrane proteins, in line with one of the talks of this morning in relation with MAM, the relation with endoplasmic reticulum, lipids, and mitochondria, that these proteins are also altered in these membranes. So this is the last one. And finally, we'd like to acknowledge all the people who has participated in this work, and the CIBERNET and other fundings, which are Lakasha and Interreg. Thank you very much again for your attention.